Welcome to Life in Biology. This is Dr. Joel Graff, and today we're going to have the slides that I drew out for a lecture given on February 14th. We discussed how the cell goes about making proteins that'll end up sticking around in the cytoplasm versus cells that are going to be secreted or transmembrane proteins, and how the endomembrane system helps accomplish the task of secreting proteins from the cell. So, without further ado, we're going to start out discussing the central dogma, which is DNA gets read and made into RNA, and RNA is read and made into protein. So we have our transcription step and our translation step. And then just to talk about where in the cell this all occurs, the DNA is obviously in the nucleus, so that when you transcribe it, it'll be in the nucleus as well. Then the nucleus has pores, and the RNA will be shuttled through the pores out into the cytoplasm. And at that point, the ribosomes are in the cytoplasm and the proteins will be made. So if the protein's destined to be just a protein hanging out in the cytoplasm, or maybe it's one that goes from the cytoplasm into the nucleus, this is the general gist of when and where those steps of the central dogma occur. But this is just half the story, and the reason for that is that we have to account for the proteins that are going to be either transmembrane proteins or secreted. And the transmembrane proteins, these might be proteins that end up on the surface of the cell, the plasma membrane, but there's also plenty of other membrane-bound organelles in the cell where if you want to get a protein as transmembrane, during its translation, you're going to be doing that work in the ER, which is the topic of the next one. So here is kind of a rewrite of what we just saw on the last slide. We have DNA transcription occurring in the nucleus. RNA will go out a pore in the nucleus, out to the cytoplasm. Then you get translation. And then the question is, how does the protein get from out in the cytoplasm to in the ER, and so that then it could be secreted? And that's what we're going to pay attention to. So you've got secreted proteins and then also transmembrane proteins. There's not a whole lot extra on this drawing, so I'll just move on to the next one. It's a far more detailed drawing, and so it'll take me a second to get through. But this is known as the endomembrane system. So DNA transcribed in the nucleus into RNA, exported through a nuclear pore, and then RNA starts to get translated. However, you start translating the RNA into protein, but there's certain amino acids that start off the protein sequence called a signal peptide, and this certain sequence of amino acids is going to be recognized by a signal recognition particle. And so the signal peptide being bound by a signal recognition particle then causes translation by the ribosome to pause. So when that RNA ribosome translation event pauses, what happens is that the ribosome will end up getting docked. That's what these red dots on the ER over here are supposed to be. You have the ribosome getting docked onto the ER membrane, and then you have an mRNA is still on the cytoplasmic side, and you have the mRNA continue to be translated at this point, and any protein, the signal peptide and any protein that follows, is going to end up being in the lumen. So this docking site is called the translocon, and it's kind of like we had a port for the mRNA to get out of the nucleus to get out to the cytoplasm. This is also a pore, so the ribosome docks right on the pore, and as it reads the mRNA and as it's making the protein gets entered in. So now that the protein's in the lumen, it can hang out there, and then at some point it might make it to part of the ER where you start having some budding of the ER membrane occurring, and when that happens, I explained it as blows a bubble. And this is called a transport vesicle that gets released from the ER. And so those transport vesicles are shown here, and they get moved, as you might expect something called transport. They end up getting moved. They move out from the center of the cell out towards the edge where the Golgi apparatus is, and another name for Golgi apparatus is Golgi body. And then those transport vesicles will then fuse to the deeper side of the cis of the Golgi. I'm going to say that that's the cis side and that this is the trans side, but I might have that backwards. But anyway, the protein will end up getting into the lumen of the Golgi at that point, and then you have more budding and fusion events, and eventually, after it's gone through the Golgi, you're going to have vesicles being released, and so now our protein is in these vesicles, and it could fuse with the plasma membrane. Not all vesicles that bud from the surface of the Golgi, the trans side of the Golgi, are going to end up getting fused to the plasma membrane, so they might get carried somewhere else in this cell to other organelles, and then they could fuse with those other organelles, and now you've got the protein within those vacuoles. Or if it was a transmembrane protein, which we haven't really been discussing at this point, but if it was a transmembrane protein, it would be in the membrane of the ER, then it would be in the membrane of these transport vesicles, and it would be in the membrane of the Golgi, and it could be in the membrane of these transport vesicles, and then when they fuse with another organelle, they end up being part of the membrane of that organelle they got recruited to. Let's see. So the reasons for the pause, I guess I kind of talked this out, but let me walk through this handwriting because it's kind of hard to read when the Smart Podium just gets saved to PowerPoint. But the first few amino acids of protein contain a special sequence. It's called the signal peptide, and I did mention that. This recruits a signal recognition particle that has the ability to recognize the signal peptide, and then translation resumes once the whole thing's been docked on the ER. So I just kind of talked through that earlier, but in class I did write that all out on the board as well. But when you get the fusion of these transport vesicles with the plasma membrane, now the protein that was in the lumen of those vesicles will end up being secreted from the cell. Okay, this is a pretty poor drawing. I was showing that you've got vacuoles in the cell that will then fuse with the cell membrane. Again, this is a bad drawing. It's probably best just to go and look this up online. I think I was trying to use color coding where I used blue being the outer leaflet of the vacuole and black to be the inner leaflet of the vacuole. And by leaflet, I mean it's a phospholipid bilayer. And so then you have two layers. Bilayer means two layers. So the outer layer was blue and the inner layer was black. When I had it fused with the plasma membrane, which the inner leaflet was red and the outer leaflet was black, I think I have my colors switched around here, but again, don't take this drawing with a grain of salt. I might have had a little bit of mistake as I drew that out, but anyway, again, yeah, these are the transport vesicles. Since this was over here, what I did is I was drawing the phospholipids and showing how they formed that phospholipid bilayer. And so that this black line on the plasma membrane is supposed to represent all those phospholipids that make up that outer leaflet of the membrane, whereas these red phospholipids is supposed to make up the this leaflet of the bilipid phospholipid bilayer. Okay. That's a bad drawing, so we'll move on. Okay, so then we went on to talking about endosome recycling. So you can have some sort of bulk transport, and we in class we talked about phagocytosis, endocytosis, and pinocytosis. So those are examples of bulk transport where something outside the cell will end up being inside the cell within a vacuole. The vacuole has a specific name of being an endosome. And as the endosome moves from the surface of the cell to deeper in the cell, it goes through a maturation process. So the early endosome is what you call it when it first gets into the cell, and then it goes deeper and deeper, and at some point it might start fusing with lysosomes. So then that'll be be called a late endosome, and the lysosomes have their own membrane. They're fusing with the vacuoles of the endosomes, and so now we've got a late endosome. And what happens is the lysosome has some proton pumps. It also has proteases and other enzymes for degrading material. And so when you get the fusion of these two things, whatever it was that was being taken up by the cell here will start to get degraded, and by the time you're in a late endosome, it should mostly be degraded. This is a way that cells can collect nutrients. So by digesting, it's almost like its own little stomach here with the acids. But you can collect nutrients, and then which are monomer. The nutrients, an example would be monomers that the cell can use to make new polymers. And there I'm thinking about the 4 categories of macromolecules we learned about: the nucleic acids, proteins, lipids, and carbohydrates. So those are the types of things that might be able to be broken down within the endosome, and then the cell could reuse that. And then whatever the cell doesn't use within that late endosome can then get discarded as this late endosome will fuse with the plasma membrane out on the surface of the cell. So that's the endosome recycling. In addition to having this practical benefit of being able to chew up things and collecting nutrients, another important point to make is that because you're going to be basically blowing a bubble into the cell to make these, so this membrane here originally was the plasma membrane. Because you're using up the plasma membrane to make these vacuoles, it's good that these vacuoles will then go out and fuse with the surface of the cell, so that then those lysosome and endosome membranes will end up out on the surface of the cell again. And the cell doesn't run out of plasma membrane because it's recycling it. Okay, a few more vacuoles or vesicles to talk about. I've been using vacuoles and vesicles fairly interchangeably. Vesicles is probably what I should have been saying. But vacuoles, they're going to be used for storage, or they might store toxic molecules. In the plant cells, we had the water vacuole. And so there's all sorts of things dissolved in the water vacuole. So anyway, that's what those vacuoles can be used for. And then peroxisomes, you should think hydrogen peroxide. And hydrogen peroxide is a molecule that is reactive. It can then be destructive. And so things that end up getting combined with a peroxisome, they'll get broken down. So for example, one reason you might break things down in a peroxisome is that in your liver, you might have the task of breaking down toxins, and so the liver is going to be filled with these peroxisomes. Okay, then we're going to switch over to, we're talking about cellular respiration at this point, and this is just a brief preview of cellular respiration. We're going to actually do photosynthesis first this year and then do cellular respiration. But, excuse me, the next organelle that we're going to look at is the mitochondria. So that brings, I'm trying to tie in the cellular respiration with the mitochondria, because the mitochondria is considered the powerhouse of the cell. So if we take cellular respiration, that's when you take glucose plus oxygen and you release CO2 and water. If you can break that down into groups of steps, we'll call it phases. The first phase is glycolysis, breaking the glucose. Second phase is deciding what to do with the pyruvate that you end up with at the end of glycolysis. This is a branch point, and so there's going to be some questions asked. What's the status of the cell? What direction do we go from here? Then we have citric acid cycle, which is also known as the trichloroacetic acid cycle or the Krebs cycle. So I will use those interchangeably, mostly because they're used interchangeably out in the real world, and so might as well get used to calling all three things all three names for this cycle. You use them interchangeably. Phase 4 is the electron transport chain, and phase 5 is ATP synthesis. Sometimes phases 4 and 5 are coupled together, and that's because the glycolysis happens out in the cytoplasm. The pyruvate gets dumped into the mitochondrial matrix. We'll talk about what that is in a second. The TCA, also the citric acid cycle, TCA, Krebs cycle, whatever you want to call it, it also occurs in the mitochondrial matrix, and then the electron transport chain and ATP synthesis occur at the inner membrane of the mitochondrion. So I was just naming all sorts of parts of a mitochondrion. That's the singular version of mitochondria. And so let's talk about what it looks like. It's got an outer membrane, which I show in black here, and then it's also got an inner membrane. And you'll notice the inner membrane has a lot more surface area because it has all of these invaginations and folds. And because of all of those folds, now we got all that membrane. So the pyruvate that was created in glycolysis in step 1 will then get shuttled into the mitochondrial matrix. That's the lumen within the inner membrane, is called the mitochondrial membrane. The pyruvate builds up there. It's 3-carbon molecule. 2 of its carbons get shuttled into the citric acid cycle. And from there, we have a bunch of electron transport molecules come into play. They're going to dump off their electrons here at these proteins at the electron transport chain, and that will cause pumping of protons out here. And once enough protons are built up out here, you're going to have the protons will flow through this ATP synthase complex, and each time the protons flow through, they turn this little propeller. I'll draw it out better at some point, but that's the ATP synthase, and the propeller gets turned, and whenever it's turning around, it's making ATP. So those are the 5 steps: glycolysis, pyruvate metabolism, citric acid cycle, electron transport chain, and ATP synthase. And this is the mitochondria. This is where it all happens. And if I didn't say it already, the space between the outer membrane and the inner membrane is called the intermembrane space. Okay, and this is just a couple of figures I found online, but I don't know if they're copyrighted, so I'll just get off of those real quick. That was the end of the lesson. This has been Life in Biology. I'm Dr. Joel Graff, and you can like and subscribe or not. Have a good weekend.